Humans have been manipulating genes for thousands of years. We just didn't call it genetic engineering back then. Think about how we got from wild wolves to the dogs we have today. Our ancestors selectively bred wolves with desirable traits, like a gentle temperament. They bred these wolves over and over, gradually shaping the species into the many dog breeds we have now. This process of selecting and breeding organisms for specific traits is called selective breeding. It is a form of genetic engineering. Farmers have used this method for centuries. They breed crops to be bigger, tastier, and more resistant to pests. This ancient practice laid the foundation for the sophisticated gene editing techniques we have today. Selective breeding is a slow process. It can take many generations to see the desired result. However, it demonstrates our long history of manipulating genes. It shows our desire to improve the organisms around us. This desire has led us to the revolutionary field of modern gene editing. Modern gene editing is different from selective breeding. It is much more precise. Scientists can now target and modify specific genes within an organism's DNA. Imagine being able to edit the genetic code like a text document. This is the power of techniques like CRISPR-Cas9. CRISPR-Cas9 allows scientists to cut and paste DNA segments, adding or deleting genes with incredible accuracy. This technology is already being used in various fields. Scientists are developing disease-resistant crops, creating new treatments for genetic diseases, and even engineering mosquitoes that cannot transmit malaria. The potential applications of gene editing seem limitless. However, this power comes with a responsibility to consider the ethical and safety implications. The ability to directly manipulate the building blocks of life raises complex questions. What are the long-term consequences of altering an organism's genetic makeup? How do we ensure that these powerful tools are used responsibly and ethically? These are just some of the questions we need to address as we venture further into the era of gene editing. The potential benefits of gene editing are vast. One of the most promising applications is in the field of medicine. Scientists believe that gene editing could be used to treat a wide range of genetic diseases. These diseases, often debilitating and incurable, are caused by defects in a person's genes. Gene editing offers a way to potentially correct these genetic defects. This could lead to cures for diseases like cystic fibrosis, sickle cell anemia, and Huntington's disease. The ability to target and repair faulty genes could revolutionize healthcare and improve the lives of millions. Beyond treating diseases, gene editing could also enhance human capabilities. Scientists envision a future where gene editing could enhance muscle strength, boost cognitive function, and even extend lifespan. However, such applications raise ethical concerns about designer babies and genetic enhancement. It is crucial to proceed cautiously and consider the potential consequences of altering the human germline. Section 4, From Farm to Table. Real-world applications of gene editing. Gene editing is not limited to the realm of medicine. It is also being used to improve agriculture and livestock. Scientists are developing crops that are resistant to pests and diseases. This could reduce the need for pesticides and herbicides, which are harmful to the environment and human health. Gene editing is also being used to improve the welfare of farm animals. Scientists are developing animals that are resistant to diseases like porcine reproductive and respiratory syndrome, or PRRS, in pigs and avian influenza, commonly known as bird flu in poultry. This could reduce the suffering of animals and improve the efficiency of food production. For example, in 2015, an outbreak of avian influenza in the United States resulted in the culling of millions of birds. This had a devastating impact on the poultry industry and raised concerns about the spread of the virus to humans. Gene editing could potentially prevent such outbreaks by creating poultry resistant to avian influenza. Such applications highlight the potential of gene editing to address global challenges in food security and disease control. Section 5, The Case for Gene Editing, a Tool for a Healthier Future. Proponents of gene editing argue that it is a powerful tool that can be used to improve human health, animal welfare, and the environment. They point to the potential of gene editing to cure diseases, reduce the use of pesticides, and create more resilient crops and livestock.
Gene editing is a more precise and efficient way of making genetic changes than traditional breeding methods. It has the potential to improve animal welfare, reduce the environmental impact of livestock farming, and enhance food security.
Supporters of gene editing also emphasize the need for a balanced approach. They acknowledge the ethical concerns but argue that these concerns should not overshadow the potential benefits. They believe that with proper regulation and oversight, gene editing can be a force for good in the world. Section 6, Ethical Crossroads, navigating the concerns of gene editing. Despite the potential benefits, gene editing has sparked heated ethical debates. Critics argue that it is a dangerous technology that could have unintended consequences for human health and the environment. They worry about the possibility of creating designer babies, the potential for genetic discrimination, and the unknown long-term effects of altering the human germline.
Gene editing is a distraction from the real issue, which is the need to move away from intensive factory farming. Instead of trying to engineer animals to fit into cruel and unnatural systems, we should be focusing on creating farming systems that meet the needs of animals.
The ethical concerns surrounding gene editing are complex and multifaceted. It is crucial to have open and informed discussions about the potential benefits and risks of this technology. Striking a balance between scientific progress and ethical considerations is paramount as we navigate the uncharted waters of gene editing.